Hello, everyone. Welcome to CyVerse's fall webinar series. I'm Tina Lee, CyVerse's User Engagement Officer. Today's webinar is on BioViz Connect, and it's a great example of how researchers can extend CyVerse for their own community's particular needs by using our APIs to access backend services, in this case, to create a custom web application for genomics, visualization, and interaction. For information on this season's webinars and past webinars, including a whole playlist of webinars on genomics and genomic file manipulation, please be sure to visit our webinars page at CyVerse.org/webinars. Most of you hopefully are aware that CyVerse is moving towards an NSF-funded cyber infrastructure project towards a sustainable enterprise, and we are supported in part by subscriptions to our platform. Our mission will continue to be to deploy a national computational research infrastructure for data-driven discovery and workforce building. And these webinars will remain an important component of that mission. So we're glad you're here. Some quick housekeeping right now, and then on to Nowlan and Karthik's presentation. Our webinar today is about 30 minutes with time at the end for your questions. Please open the Zoom chat window and type your questions there. All of our webinars are recorded, and I will post this video recording on our website webinar page in a day or so. We now have more than 65 webinars organized into multiple playlists on science, technology, and CyVerse platform topics to help you learn and do your science at your own pace. So now I will introduce our presenters. Nowlan Freese is a postdoctoral research associate in Dr. Anne Lorraine's Genome Visualization Lab at UNC Charlotte's Department of Bioinformatics and Genomics. Nowlan focuses on improving genomic visualization through tools, including BioViz Connect and the Integrated Genome Browser. His research interests include how intragenic methylation controls alternative splicing in rice, and how methylation can make rice more resistant to abiotic stress. Karthik Raveendran is also a member of Dr. Lorraine's lab and is a doctoral student in the Department of Computer Science. Karthik researches visualization methods as applied to bioinformatics and genomics and helps build tools and develop techniques for visualizing genomic data. So welcome, Nowlan, and welcome, Karthik.
Yeah, thank you, Tina, and thank you everyone for being here. So I'm Nowlan, and today I'll be telling you about our project, our web application, BioViz Connect, and how you can use it to view your CyVerse data within the Integrated Genome Browser. Make sure you do this. So our group is at UNC Charlotte, and we have been developing the Integrated Genome Browser. The Integrated Genome Browser project, or IGB, is free and open source. So if you want to try it out or you want to use it, you can just go to our website, BioViz.org. There's an install IGB link, and then we support Mac, Linux, or Windows, and you'll install it on your laptop or desktop. So it's a run locally. So what can the Integrated Genome Browser do for you, or what can you do with it? So IGB can load and view all of the common bioinformatics or genomics file formats. So if you've done something like a ChIP-seq experiment, RNA-seq, whole genome bisulfite, or like a resequencing project, you should have a line sequence file. So these would be like BAM or SAM. If you've done like an annotation project, you might have something like a bed file, GFF, or GTF. We can view those, or something like a coverage graph. So these would be bed graph, wiggle, bigwig, etc. We can view those as well. Now, one of the main features, or what we're proud of with IGB, is the ability to interact with and kind of do some visual analytics on that data. So I have an example here, just some images from IGB. This is from a mouse experiment looking at ChIP-seq data for SOX9, or the transcription factor SOX9 and binding. So what I've done here is I've kind of layered that data within IGB. So I have our ChIP-seq data on top, and then below that, we have our mouse reference annotation. And this kind of looks like a barcode or something, and that's because we're actually zoomed way out. So we're looking at kind of like the whole chromosome here. And what I've done with IGB is I've actually used it to color code my ChIP-seq data. so that the high scoring peak regions are highlighted in red. So you can imagine, you're looking at the whole chromosome. You might find some peaks of interest, or maybe you have a gene of interest. So then you can zoom in on that region. And in IGB, you can actually set a user-defined thresholds. You kind of do this interactively, and use that to highlight some peaks of interest. So here we've highlighted, maybe it's a binding location that's particularly prominent, and then we can actually zoom in even further. So down to the level where we're looking at the sequence as well as the beginning of the gene annotation. And you can use IGB to search for motifs. So you can actually search the sequence. And so maybe there's a motif that we're aware of, and we want to see, does that fall within our ChIP-seq binding location? And in this example, of course, it does. And so that works out great. Now, of course, the other kind of main use case for IGB is creating figures for publications, presentations, etc. So I've just pulled out a couple here from recent publications. And you can kind of see how other groups have used IGB to create figures for those publications, doing things like using color to kind of differentiate between different samples or different types of studies. So we've got like ChIP-seq data here. So again, just kind of as an example. Now, I hope I've kind of convinced you that IGB could be useful for your work or research, but we're always interested in making IGB easier to use, more powerful, trying to add new features. Now, in the last 10 years, with the decreased cost of sequencing, we've seen the amount of data generated just grow exponentially, as well as the number of reference genomes that are coming out. And so you can imagine if you're doing something like a RNA-seq experiment, and you might do that on 10, 20, 30 samples, and you might generate hundreds of gigabytes of data. Now, IGB runs on your local machine, and you probably don't want to store those hundreds of gigabytes of data on your laptop, for example. You're probably going to want to store it on some sort of cloud data storage provider, something that's safe, secure, but also provides access to that data as easily as possible. And so our question became, what resource or what kind of cyber infrastructure provider can we try to connect with IGB? Of course, we're in a CyVerse webinar, and so that cyber infrastructure provider is CyVerse. So CyVerse has been around for quite a while now and has done a really great job of tackling a lot of these big data issues. So they provide a lot of data storage as well as computation, so the ability to run analyses on that data. And in the last couple of years, they've expanded their APIs. And I'm not going to get into APIs myself. I'll leave that for Karthik to talk about later in the talk. But because they spent that time expanding those APIs, we as an external developer, so we're not associated with CyVerse, we're at University of North Carolina, Charlotte, we were able to use those APIs to build BioViz Connect. And that's kind of like a middleware that sits in between CyVerse and the Integrated Genome Browser. And so what is BioViz Connect? So it's a web application, so it does run in your web browser. The project is free, so you can go to BioViz.org and click on BioViz Connect to try it out or to use it. And it's completely open source, so all of our source code is available. You could try it out if you wanted or take a look. The focus of BioViz Connect is to make it, again, as easy as possible to view your CyVerse data within the Integrated Genome Browser. So at this point, I'm just going to go ahead and jump into a demo so we can show BioViz Connect and how it works with IGB. So I'm going to go to our website, BioViz.org, and then I'm going to click on the BioViz Connect link. And then I'm going to click on sign in with your CyVerse ID. I want to point out that, so the login is handled through CyVerse. You use your CyVerse credentials. So you don't have to make any new login with us for BioViz Connect. Everything's handled through CyVerse. And we don't handle your CyVerse credentials itself. So this is similar to the single sign-on that you might have seen for something like Gmail or Facebook. So I'm going to go ahead and log in. So CyVerse will go ahead and authenticate me. And then once it's authenticated, we should be directed to our website, BioVizConnect. Okay. So this should look familiar to anyone who's used any kind of cloud data storage provider, anything like Google or Dropbox. And especially so if you've used the most recent version of the discovery environment through CyVerse. So what we've done here is we use those APIs from CyVerse to actually pull in all of your files and folders. So you can see everything that I have in my CyVerse account here. On the left, we have some shortcuts. So this is my home folder. You can also see all of the data that has been shared with you from other users, as well as the public data, files and folders within the community folder. So today, the data I want to look at are in our home folder. And the example I want to do today is from some RNA-seq data. And we're going to run through kind of two scenarios to give kind of an idea of what we can do with BioVizConnect and IGB. So I'm going to jump into my data here. Again, navigation is just like any other kind of cloud data storage provider. So here's the data I want to take a look at today. And there's kind of two things that might jump out at you. The first is the big green View in IGB button. So of course, again, we want it to be as easy as possible to view your data from CyVerse in IGB. The other thing is that the file names here are a little cryptic. So these are pulled from the sequence read archive. So these are SR run numbers. So they are unique, but they're not particularly human readable. So what we've done is we've added the ability to look at metadata and added edit metadata for these files. So I'm going to right click. I'm going to select metadata. So this sample is a watered control. And I have some IGB specific metadata that's been associated with this file. So it's from the Arabidopsis thaliana, or the alignment is for the Arabidopsis thaliana genome. You can see all of the genomes that IGB has access to here. I've also color coded it. So this particular one is blue. And then I have some additional information, comments that we can leave below here. Now, in this example scenario, we are interested in a file that seems to be problematic for some reason. So it's this one down here. This file is also a control, a watered control. So it should be very similar to this other sample, this watered control 1 sample. So I want to go ahead and load these files in IGB and take a look and see if we can determine what is going on here. So all we need to do is click on View in IGB. And I'll do that for the second file as well. And what BioVizConnect is doing is it's doing a couple different things. So it's first going to check and make sure that IGB is running, and it is. It's also going to check that these files are public. So right now, files do need to be public in order to be viewable. And so if we want to check that, we can right click and select Manage Link. And we can see that this file is currently public. We could remove that link if we need to. We can make the file private. And as long as it's public, we can actually copy this URL and share it with collaborators as an example. Okay, so why don't we jump over to the Integrated Genome Browser and take a look. Okay, so both files have been added as new tracks. We haven't loaded any data yet. That's why this appears gray. I just want to point out that I have already loaded the Arabidopsis thaliana genome. So it's over here. We're looking at chromosome 1, and I've zoomed into my gene of interest. Now, IGB is very interactive, so we could zoom out to the level of the chromosome if we wanted to, or all the way into the level of the sequence itself. But in this example, we want to take a look at our gene of interest. So let's go ahead and load our data. Okay, so the watered control 1 sample, this looks pretty good. So again, this is RNA-seq data, so we would expect to see lots of read coverage across the exons. So here is our gene annotation model. The thicker black bars are exons. The thinner arrowed lines are introns. So we've got good read coverage here. I would say overall this coverage looks pretty good. Whereas in our problematic sample, we do have read coverage, but the reads are highly stacked on each other and somewhat sparse. So probably an issue with either library preparation, maybe some kind of PCR duplication issue, or potentially with alignment. But I think the main point here is that we probably wouldn't want to do any further analysis with this sample without digging in and figuring out what went wrong and trying to correct it. Okay, so that's the first scenario. And again, this is kind of like a quick sanity check. So the ability to look at your data and say, does this look good or not? Now, this is RNA-seq data, so probably what we're really interested in is looking for differential expression. So let's jump back to BioViz Connect, and we will load some additional files and take a look. I'm going to switch back. And I need to check and make sure that I'm selecting the right files. So I'm going to do right-click metadata. And this is a control. Okay, that's what I want to look at. So I'm going to click View on IGB. And then I want to find my heat-treated sample. It's right here. So again, I'll click View on IGB for that one. And now we can jump back to the Integrated Genome Browser and take a look. Now, while those files are loading, I'm going to go ahead and get rid of these two watered control files. Okay, so those are both loaded. So I'm going to go ahead and click Load Data. We'll give that a second. Okay. Let's say off the bat, this looks pretty good. We've got great read coverage across the exons. These files look pretty similar. So, but IGB is only going to show us by default the first 10 or so layers of reads. So we want to look at all the reads and see if we can identify any differences in differential expression. So I'm going to click this button up here. This is a kind of shortcut button to show us all of the available reads for both of these samples. Okay, so now we can see that, yep, we've got good read coverage here, quite a few reads. Maybe it looks like a little bit more in the heat treated versus the control. We can actually use IGB to drag click if we wanted to, and we could select a stack of these and say, okay, I've got 392 there. How many do I have here? Something like 209. Of course, that's not the most accurate way of doing it. We can also create coverage graphs in IGB through our right-click menu options here. But even that probably wouldn't be the most accurate or best way to do that, because trying to compare between samples, really what we need to do is create scaled coverage graphs. These two samples were sequenced to different depths, so we would need to account for that. Now, again, IGB can create coverage graphs, but scaled coverage graphs are a little bit more intensive, because we need to actually pull in that entire file, and we need to analyze that entire file in order to be able to create that scaled coverage graph. So that's something that would be great to run in CyVerse using their computational power. So why don't we jump back to BioViz Connect, and we can create some scaled coverage graphs. I'm going to switch. And I'm going to right click on my control sample, and I'm going to select Analyze. Now, CyVerse has access to hundreds, I think thousands of different apps that can allow you to analyze files in different ways. What we've done is created a subset of those apps that we thought would be particularly useful for anyone who would be wanting to visualize their data in IGB. So in this case, we have the option of creating a coverage graph or a scaled coverage graph. And so I'm going to go ahead and click on Make Scaled Coverage Graph. And all we need to do here is put in an analysis name. So this is like the job name and then the name of the output file. And I'm just going to do some copying and pasting here. So I'm going to use the SRR unique number, and then I'm just going to call this scaled. And then I will do the same thing. I'll put the same thing as the output file name. Then we can go ahead and click run analysis, and that job will be submitted. And then we just have to do the same thing for our heat treated sample. I'm going to go to analyze, make scaled coverage graph. I copy the name, scaled, and then I will copy that as the file name. Okay, then we can go ahead and click run analysis. So both those jobs have been submitted, and if we want to take a look at their status, we can go over here to analysis history. And here we can see history of all the jobs submitted. Our 2 jobs are currently running, that's great. Now, how long these jobs take to complete will depend on the size of the file, the type of job, so in this case, make scaled coverage graph, as well as the number of jobs other CyVerse users are submitting. So if there are a lot of jobs being submitted, you could be towards the back of the queue. So in the interest of time, I'm going to jump back to my folder where I have already completed creating some scaled coverage graphs. So again, I want to make sure I'm looking at the right file. So I'm going to right click, select metadata. We've got our control. So I'm going to click view in IGB. And then I will do the same thing for our heat treated sample. And then we can jump back to the Integrated Genome Browser. And while those are loading, I will get rid of our reads here. And we'll give that a second to load. And again, the time at which this takes to load into IGB will depend on the type of file, the size of the file, your internet speed, and how much traffic CyVerse is getting. But usually things are pretty quick. Okay, so both those have been added, so we can go ahead and click load data. Give that a second. Okay, so initially these look like they are pretty similar, but if we look at the y-axis, these are on 2 different y-axes, so we need to put them on the same y-axis. So in IGB, we can do that by going to graph. These are both graph files, and we can select both of them by hitting this select all. And then under y-axis scale, we can set that scale to a value, and I'm going to say 30. I'm going to click enter. And so now they're both on the same scale, and we can see that it looks like our heat treated sample has a lot more expression than our control. So a quick way to do a visual analysis or visually say, it looks like we've got some kind of differential expression happening here for our gene of interest. So hopefully this was a kind of good quick demo of how you can use IGB and BioViz Connect to visually analyze your data. I'm going to jump back to BioViz Connect now, and I just want to point out that all of this data plus more are available for anyone who has a CyVerse account to take a look at. So if you go into the community folder, we have a folder here called BioViz. And the data from today are in RNA-seq, Athaliana, and then our SRP project number. And so we have all of the reads as well as unscaled and scaled graphs for the samples we looked at, as well as additional. And now I'm going to jump back over to our slides, and Karthik, I will let you take over. Karthik's going to give us some more detail about how we actually built all of this.
I hope everyone can see my screen.
Yes, looks good.
Hi, I'm Karthik. Now that you've seen the BioViz Connect demo, I'm going to talk about how we built BioViz Connect. This part of the presentation is for anybody who wants to create a custom web app using Terrain API. So when a user navigates to BioViz Connect, the user, as you saw in the demo, the user is briefly redirected to CyVerse CAS authentication. CAS is Central Authentication Service, which is the login page that you saw. And the user will add their CyVerse credentials, and CyVerse will authenticate the user and send us an access token. And from then on, BioViz Connect will use that access token to communicate with CyVerse on the user's behalf. We don't have the user's credentials at all. So once we get the access token, the user is redirected to BioViz Connect. BioViz Connect is built on top of Django framework, which is a Python-based web framework. And the code that we built on top of Django helps us communicate with Integrated Genome Browser as well as CyVerse. But the user interacts with the user interface, which is built simply with HTML5, CSS, and JavaScript. And this whole infrastructure is hosted on Amazon EC2 instance. Now, BioViz Connect communicates with CyVerse using Terrain API. For those of you who doesn't know what API is application programming interface, which is essentially a software tool for two applications to talk to each other. So in this case, it's BioViz Connect and CyVerse. So BioViz Connect will send an API request to Terrain, and Terrain API will send an API response. Now let me show how a Terrain API request looks like. So this is what it looks like. A Terrain API request looks like a web URL. So, and the response looks like this. Now, because this is a web URL, I can actually show this in a browser. So if I add this request here, CyVerse will send a response, and the response here is that I'm not authorized because I'm not sending CyVerse my access token. So I'm gonna make an API request that does not require an access token, just to show how the response looks like. Yeah.
Karthik, excuse me, could you, I was gonna say increase your screen just a little bit, but I see what you're trying to prove. Sorry about that.
Okay. Yeah, so yeah, this is the response that we get. So this is, I think, all the public apps available in CyVerse. So let me just go back to the presentation. So we make a similar request when the user first logs into BioViz Connect. We make a similar request to CyVerse, that is this request, and what we get is the users, a list of users, files, and folders, and this is what the response looks like. And then this response is translated to our BioViz Connect user interface that looks like this, and this API request is customizable. So we can actually, if you don't want the entire list of files and folders of the user, we can ask CyVerse to send us a limited version of that. Let's say we need the first 100 files and folders and the next 100 and so on. And we use that feature to paginate this table. So the first page will have 100 files and folders, and the next page will have the next 100. We can also ask CyVerse to sort these folders and files and send it to us. So that is, these two features are very helpful as a developer because we don't have to think about that when we're developing these things. So CyVerse APIs help in that regard. So using APIs like this is how BioViz Connect communicates with CyVerse. Now I'm going to talk about how BioViz Connect communicates with IGBee using its own collection of APIs. So if a user clicks on view in IGBee, a public link is sent to Integrated Genome Browser, and the link is for that particular file. So when a user clicks on that, this is what the link looks like. And CyVerse calls it a public link. And that's because in CyVerse, users can share their files with other CyVerse users internally, but externally, the user has to use a public link to share the file. And sharing the public link is not enough. The user has to provide a public link permission as well. And in BioViz Connect, you can control that permission in the UI itself. So the permission looks like this. So for this file, the permission, the read permission is provided to a user called anonymous. So if you give permission to anonymous, this this public link will be valid. If you remove that, it will be useless. So if a user wants to send, along with the public link, if the user wants to send like IGB related metadata, some details to IGB so that the user can view it in their own style, they can use this API request. So this is the metadata API request that CyVerse provides. So for each file, you can add metadata based on one particular app. So in this case, we can ask CyVerse to send us the metadata of a particular file, providing the file ID, and we get a response like this. And out of that response, we filter out the integrated genome browser parameters, and we translate that to our user interface. And the user can change those parameters. And this when the user clicks on view in IGB, along with the public link, we send this data as well. Now, one of the important use cases for BioViz Connect is using the apps in CyVerse. So we have a curated list of apps in CyVerse for IGB. So what we've done is we created an integrated genome browser community, and we've selected a few apps and mapped those apps to that particular community. So if you use this API request with the community name Integrated Genome Browser and /apps, we get a list of all the apps that is mapped to that particular community. And we use this response to create a user interface. Now, when a user clicks on one of these apps, we send another API request with that particular app ID. And with that response, we create a user interface. Now, how we can do that is because when we do this API request, when we actually make the API request, CyVerse, so this is what a CyVerse app looks like in CyVerse. So CyVerse will send all these details with that response, like the text fields, what input type should be entered, how can we validate that particular input. All that will be sent to us, and we use that to create this user interface. So this is how BioViz Connect communicates with CyVerse and how we run all the functions. So our code is available in Bitbucket. So if anybody wants to build their own custom web app using BioViz Connect code base, they can. And our deployment strategies are also available there. This is the team that built BioViz Connect, and I would like to thank the CyVerse team as well, because they helped a lot whenever we hit a roadblock. And these are the Bitbucket resources and some other links that for the genome visualization lab in UNC Charlotte. So that's it. Thank you.
Thank you, Karthik. Thank you, Nowlan. This has been great. And this resource page is very helpful. I will post this slide presentation as well on our webinar page. We did have one question that Nowlan has also answered with the links in our chat, but just so for the video recording, somebody would like to know if there's an FAQ for IGB and how it's evolved and why it's evolved. And so Nowlan pointed them to https://wiki.bioviz.org/confluence/. And so that's obviously here on this resource guide too. So we will include those, like I said, on the webinar page. My question, because I'm always interested in what you guys are doing, is what's on the roadmap for future functionality or anything, if you can share that with us, if it's not top secret.
Yeah, I mean, I think, so one of the things that we're always having to do, or I guess, so, as CyVerse continues to expand the Terrain APIs, anytime they add additional functionality, that kind of opens new things to us. So I think there's kind of new things we can do and we need to look at to make BioViz Connect even better. I think adding additional apps is definitely on our roadmap. Again, we created kind of this small subset to begin with of curated apps, but we could add a lot more based on what we think people would find useful. And I think those would be kind of the two main things that are directly in our roadmap within a short period of time.
That's great. I know it's a lot of work. Karthik, you made it look easy, but I know it's a lot of work. Well, thank you, gentlemen. If there are no further questions, we really appreciate you guys sharing your expertise and knowledge in developing this. And we thank Dr. Lorraine also for letting us invade your time and share your skills with us. Please do join us for next month's webinar when Dr. Bonnie Lafleur and Dr. Xiao Sun will introduce strategies for benchmarking for novel pipeline development in single-cell RNA-seq experiments. That's going to be October 21 at our 10 p.m. Pacific time slot. And until then, thank you again to the Lorraine Genome Visualization Lab. And take care, everyone. We'll see you next month. Thanks, Nowlan. Thanks, Karthik. Take care.